Fab. I think we'll get going there. So welcome everybody to, like I said, our fifth and final instalment of the immunoassay week. This week, if you're new to joining us, we've had live workshops every day going over the best practices for different immunoassays. So my name is Dr. Amy Turner. You may have seen me giving the Western Blot workshop on Wednesday. If so, hello again, welcome back. I'm the scientific liaison here at Proteintech, and I will be chairing this workshop today. I'm really excited to have Dr. Poppy Nathan with us today. She has also been chairing the session, so she'll be back to give her own instalment of how to do ELISAs. So best practices for reliable data is what she will be focusing on today. We'll have a Q&A session at the end, and I'll be answering questions throughout. So please just feel free to pop any questions you may have in the chat box so that everybody can see them, and we'll hopefully get back to you at the end. But just before we start, I'd like to give a very brief introduction to Proteintech in case you are unfamiliar with us. So first off, if my slide changes, there we go. So Proteintech is predominantly a life science reagent company. Our product lines are comprised of 4 key portfolios. So our first and our namesake is Proteintech. This is where all of our antibodies reside. So antibodies and immunoassays are our bread and butter. And our CEO, he has a big goal of targeting the entirety of the human proteome with our antibody reagents. So there is about 21,000 human proteins, and at the moment we have antibodies covering over 14,500 of those targets. These are in polyclonal, monoclonal and recombinant formats, and we also have kits that are derived from our antibodies in this portfolio as well. So IHC kits, ELISA kits, cell separation kits and antibody labeling kits. Secondly, we have our humankind range. So this is our portfolio of cytokines and growth factors. So different to other players on the market, our humankind cytokines and growth factors are all produced in human cells. So this is compared to other players on the market who might produce these proteins in E. coli or insect cells. Now, because they are produced in human cells, they have their native folding and post-translational modifications, which means they're really highly bioactive and fantastic for things like cell signaling research or stem cell and gene therapy. Anything that kind of uses cell culture, these proteins are fantastic for. We have them from research use only all the way up to GMP grade. So if you are lucky enough to be in a project that goes from bench side to bedside, then these products are really useful for those. Thirdly, we have our Chromatech range of nanobody-based reagents. So if anybody has seen our mascot, our alpaca, the reason we have Alice the alpaca as our mascot is because alpacas, camels, llamas and other camelid species, their immune response raises heavy chain only antibodies. We take the variable domain of these antibodies, and that is what is known as a nanobody. So it is a single polypeptide small proteins. It's about 10 times smaller than your traditional antibody. And these reagents are fantastic for things like immunoprecipitation because they can withstand harsh buffer conditions. And because they're so small, they're fantastic for super resolution microscopy. Finally, Proteintech also has our Proteintech Genomics portfolio, which is a portfolio of cocktails of DNA conjugated antibodies for use in single cell RNA sequencing pipelines. So you can essentially profile at the single cell level, both transcriptomic and proteomic data. So this is a really exciting product line that is our sort of most recent one. So we are a company kind of for scientists by scientists. So all the way from the top, our CEO was a professor of immunology, and we have PhD level trained researchers throughout all of our teams within Proteintech. Like I said, all of our products are manufactured by us, so you cannot buy any of our products elsewhere. That means that if you have any issues with any of our products, you can essentially get in a direct line of contact, essentially with the scientist who made that product, and we can give you troubleshooting advice tailored to your project needs. This has allowed us to actually receive the Researcher's Choice Award by CITAB recently, and that's also owing to the fact that we have the largest in-house portfolio of antibodies for the human proteome, like I mentioned in my previous slide. We sort of measure our success by the success of our researchers as well, and recently we have hit a milestone of over 350,000 global product citations, which has boosted us into the top 5 globally cited antibody companies. So because we manufacture everything ourselves and we do have some fantastic researchers who work with our products, what we hold in high stead is the fact that everything is open access. So if you have any sort of product validation data that you would like from us that isn't currently accessible on our website, then please do feel free to get in touch. Our outstanding PhD level technical support will be more on hand to help you with any sort of issues that you may be facing. And we do offer global next day delivery with our 7 inventory sites, manufacturing and research sites on 3 different continents. What I'd like to highlight before we get going with the ELISA talk is actually one of the couple of new first in industry developments that Proteintech have released this year. So firstly, we have a 3D epitope viewer. So when I think back to my PhD days, the only information that I could get from my antibody suppliers was, does this antibody bind to this protein? Yes or no? And that was it. Well, Proteintech have kind of gone a step further, and we have essentially mapped, experimentally mapped, where exactly our antibodies are binding to our protein targets, and retrospectively mapped that back onto a 3D structure, which you can now see as a tool on our website. So you can find your favorite protein, spin it around, zoom in, find the domain that you're particularly interested in, and see if we have an antibody that binds to that particular site. So at the moment, we have over 2,500 of our proteins mapped, and this is an ongoing project, and we hope to do this for the entirety of our portfolio. For those that we haven't mapped already, we still have our immunogen selector module as well. So if you consider that you've got one long protein, your amino acid sequence, you know the sequence of it, you have a particular domain of interest, you can toggle this immunogen selector module to that particular domain of interest, and it will filter for the antibodies that you would be useful in your workflows. Secondly, we've also released very recently the first in industry able AI-powered search tool. So this is an AI-powered search tool that's on our website that can help you to pick the antibodies or reagents that are most suited to your needs. So you can ask it for tailored product recommendations, for example, find markers for subcellular organelles. It will offer step-by-step experimental guidance and save you valuable planning time, reducing trial and error. I always think if your PI is really busy, you can ask Able and it can kind of help you out there. One of my favorite things about Able as well is that it understands and responds in multiple languages. So it is really democratizing the sort of global research that Proteintech can help our scientists do. So without going any further into Proteintech, I would like to introduce our speaker for today's talk. So Poppy completed a Wellcome Trust funded PhD at the University of Birmingham, where she was working on immune cell trafficking in the context of chronic inflammatory disease. Her PhD focused on investigating the mechanisms of action of an immunoregulatory molecule with the goal of developing a therapeutic to reduce inflammatory symptoms in patients with type 1 diabetes and rheumatoid arthritis. She's frequently used ELISAs in her research, so that's very useful for today's session. And she was using it to investigate the changes in inflammatory markers in biological samples. Poppy is now our wonderful scientific marketing specialist at Proteintech. She delivers immunoassay training workshops all the time. She creates educational content that resonates with Proteintech's diverse research audiences, including detailed product guides, blog posts, protocol videos, and online training courses. So with that, I'm going to pass over to Poppy, where she's going to talk to you all about ELISAs.
Thank you, Amy. Thanks for that great introduction. And hi, everyone. It's great to be back as a presenter today. So I'll just share my screen there. Amy, if you could just let me know if you can see that all okay.
Yeah, all good.
Perfect. So today I'm going to be talking all about ELISAs, and hopefully, importantly, I'm going to have some tips for you that are going to help improve that assay reproducibility and really help you get that reliable data. So I'm going to start off with a quick introduction to ELISAs and walk through each of the individual steps of the workflow. We'll then cover some tips and tricks for best practice before ending on some data analysis and then hopefully some more complex troubleshooting towards the end. But first of all, what are ELISAs? So ELISA stands for Enzyme-Linked Immunosorbent Assay, and they're assays which work by immobilizing an antigen-antibody complex to a microwell plate, and enzymes that are then attached to that complex catalyze a reaction to result in a colored product. They're considered to be the gold standard for quantitative analysis of quite crude biological samples. So for an ELISA, you can use things from cell supernatants, you can run human serum, CSF, you can use real biological samples. They're also fairly quick, they're simple, they're high throughput, and they're easy to automate. But however, it's worth bearing in mind that ELISAs can't be used for spatial information. So if you do need some information or you want to look at protein localization, for example, then you might want to use a different application like immunohistochemistry. So they're widely used both in research and in clinical settings. So ELISA formats are what you'll find in your LFT tests for things like COVID. They're also used clinically to detect for things like HIV. So two of the most common protein quantification applications in the lab are your ELISA and your standard Western blot. But how are they different and when would you use each one? So they both allow for very high sensitivity detection of protein or analyte. ELISAs can be quantitative as well as semi-quantitative, whilst Western blots are more qualitative. The biggest difference, however, is what I've already mentioned, that ELISAs can be used with quite a crude sample preparation, whilst for a Western blot, you'll have to make a protein lysate, you often have to denature and get a full linear protein. One consideration for ELISAs is that they will only tell you the concentration of your target, whilst Western blot, again, will let you find out a little bit more information, things like post-translational modifications, relative expression, phosphorylation, mutations. You can look for those with your Westerns. So there are actually 4 different types of ELISAs, which have different capture and detection methods. So your direct ELISAs work by immobilizing the antigen of interest directly to your microwell plate, and then using a directly conjugated primary antibody for that detection. Indirect ELISAs are very similar, except you have a conjugated secondary antibody to amplify the signal. Sandwich ELISAs are the only ELISA that utilizes an antibody pair. So rather than immobilizing the antigen directly to the plate, you immobilize a capture antibody, which then captures your protein of interest from your sample and your standards. You then have a detection antibody that also goes against that antigen. And then you have possibly a secondary antibody or just a biotin conjugated detection. You then also can get competitive ELISAs, which quantify the amount of analyte in a sample by measuring how that interferes with an expected signal. However, when most people talk about ELISAs, particularly in a lab setting, what they're referring to are these sandwich ELISAs, and this is what we're going to talk about today. And that's because sandwich ELISAs are considered the gold standard of ELISAs. As they're the only ELISA that utilizes this antibody pair, it can really increase the sensitivity and the specificity up to around 6 times compared to a direct ELISA. Because you need those 2 antibodies to recognize your antigen, you're going to get a much higher likelihood that what's bound is binding your protein of interest as opposed to background or off-target effects. So going a little bit more detail then into the sandwich ELISA process. The first step is to coat that plate, as I said, with a capture antibody. It's then important that you also coat the plate with a blocking agent to block any remaining binding sites and prevent that unspecific binding. You can then add your samples and standards, and your analyte of interest will be captured by that capture antibody. You can then add the detection antibody, which, as I mentioned, will either be directly conjugated itself, or you can amplify the signal with a secondary. But whichever method you use, the detection antibody usually will be biotinylated, because that binds with very strong affinity to streptavidin, which allows for streptavidin HRP to be added to catalyze a reaction with the substrate, resulting in that colored product. But if we go over each of these steps in a little bit more detail now, before you even get onto coating your plate, you really want to consider what your plate layout is going to look like. So what's really important in an ELISA is that you run all of your samples and standards in replicates. So you should be running these in at least duplicates, but if you have enough sample, and let's say maybe you're fairly new to ELISAs, then you might want to consider also running in triplicates. It's also really important, as with all immunoassay applications, that you include your appropriate controls, and we'll come on to those in a little bit more detail on the next slide. One thing as well with ELISAs is before you've started your experiment, once you've got your plate layout, you know exactly how many samples you're going to be adding, how many standards you're going to be adding, how many replicates. You want to be working out your sample and reagent volume needs ahead of time. And that's because Something that I hope will come up a lot during this workshop is the idea that ELISAs are simply a chemical reaction. So that means that they are significantly affected by things like temperature and incubation time. And if you get halfway through your plate during one step and realize that you haven't made up enough wash buffer or you haven't made up enough detection antibody, the time it's going to take you to prepare more of that is really going to impact your results. And again, that's what's going to affect the reproducibility of your assay. Make sure you record at this point any sample dilutions that you want to use. Make sure you save unused strips, put them back in the packet that they came in and put them in the fridge, and they'll be good for up to a week. So as I mentioned, whenever you're using an antibody-based application, you always have to spend a minute thinking about the appropriate controls for your experiment. So importantly, you're going to need to include some kind of negative control, also referred to as a blank. And this is a well that will have your sample diluent, but no sample and no standards. So it's just reading any background noise that you're getting from the sample matrix. However, you actually want to consider what the background means for your specific experiment. So if your sample type is cell culture, a cell culture supernatant, then actually a good negative control or blank reading for you might be the cell culture media itself. You can also include chromogen blanks. So these are control wells that only have the substrate and stop solution, and this will allow you to see any reading that's coming out just from background of the substrate and the stop solution. It's also important that you add in a positive control. And for ELISAs, your standards do act as a positive control. However, if you're using a particularly crude sample matrix, then you also might want to think about adding in a spiked sample matrix to see whether or not you're getting any incorrect reading, or if the signal is being blocked from something else that you're finding within your crude sample. There is another form of control you can perform as well during the plate reading step, which is called wavelength correction, but you don't need to worry about that at this stage, and we'll come on to that when we're reading our plate. So the first step of an ELISA is, as I say, coating that plate with your primary antibody. So many kits, and all of Proteintech's kits, will come with the plates pre-coated. Not only does this save time for you on overnight incubation, the reason we do it is because it really increases that inter-assay reproducibility. If you are performing this step yourself, then it's really important to make sure that your plates that you're using are suitable for antibody binding. ELISA plates are usually hydrophobic polystyrene surfaces, and they immobilize the antibody via passive adsorption. Because that's a non-specific interaction, you need to, as I say, make sure that those plates will bind, but you also need to make sure that you're blocking those plates effectively. If you don't block your plates effectively, then any other protein, whether that's proteins found in your sample or whether that's your detection antibody, they will also bind the plates in that passive manner, and that's going to lead to really, really high background. So we always recommend, if you are blocking your plate yourself, make sure you're using a higher volume than the reaction volume. So normally, let's say you're only coating your plate with 100 microliters of primary antibody and 100 microliters of your samples and standards. You want to be blocking with about 400 microliters of blocking solution. BSA is a very popular protein block, but make sure that if you're using a kit, you are following the manufacturer's recommendations regarding all buffer composition. Some kits, for example, will recommend that you block with serum. So if, like I mentioned before, you are using one of Proteintech's ELISA kits, or you're using an ELISA kit that comes pre-coated, this is actually where your experiment will start from, because those the kits will come with plates that are pre-coated and pre-blocked. So at this point is where you can think about adding your standards, adding your samples, and at this point you also really need to take several steps to ensure accuracy. So the way ELISA measurements work is that you're going to run a serial dilution of known standards, and those standards will be at a known concentration. From those, you will create a standard curve from which you will extrapolate the measurements of your unknown concentrations. Therefore, if your standard curve is not accurate, your results won't be accurate either. So first of all, make sure you're following the manufacturer's recommendations when it comes to reconstituting your standards. And you should always try and avoid freeze thawing with this as well. If you are using a big kit and you've got a lot of standards, make sure that you're aliquoting. And something that's really important is to make sure that you are using fresh standards every plate. I've written there every time, but what I mean is every plate. So even if you're running 2 or 3 plates in one go, you need to run a fresh standards and a fresh standard curve on each plate. In order to produce that reliable standard curve, we also ask you to please don't expand the suggested range. So again, if you're using a protein standard or if you're using a kit, that will give you recommendations on it as to the highest concentration down to the lowest concentration. Those will have been extensively quality controlled in-house. And sometimes if you go above or below what they're recommending, then actually the antibody binding won't carry on how it's predicted. And so expanding the curve is not going to change the actual binding characteristics of the antibody in that specific setting. Make sure that you're diluting your standards and your samples in the same dilution buffer. As because the refractive index needed for when you do your absorbance readings has to be the same between your buffers. And also antibody binding can be affected by the sample. This is everything that's in your sample except the protein of interest. So you need to make sure that that's not affecting it as well. So the important thing to bear in mind with your detection antibody is that it needs to be able to recognize a different epitope on the target antigen. So I'll come on to antibody pairs a little bit later in this talk, but if you just think about your protein of interest, the places where the capture and the detection antibody bind are going to have to be different and non-overlapping regions, because they both need to be able to bind to the protein at the same time without getting in each other's way and without causing steric hindrance. So the detection enzyme substrate reaction can vary between kits, but very frequently, and in the case of all Proteintech kits, we use streptavidin HRP. So in order for the streptavidin to bind, the detection antibody will often be biotinylated. So the biotin streptavidin interaction is actually one of the strongest naturally occurring interactions. And it's, as I say, it's used frequently across different ELISA kits. So you then need to add a streptavidin enzyme conjugate. Again, this can vary between kits, and if you're making it yourself, you might want to spend a bit of time just thinking about this. But a very commonly used one is horseradish peroxidase. So HRP is an enzyme that's found in the roots of horseradish. It catalyzes the colorimetric reaction in the assay. We use it at Proteintech because of its rapid kinetics, makes the reaction really fast. And when you're using HRP, then the complementary substrate is TMB, and that's what you'll use. It is worth noting that HRP can be degraded by microorganisms and antibacterial agents, so make sure you're keeping your work area nice and clean. Also, the enzyme can be inhibited by cyanides and sulfides, and we also recommend that you avoid placing this in direct light, so just be careful with how you're storing and using these reagents. So once your detection antibodies on, you can now add your substrate solution. So as I said, for HRP, this is often TMB. You want to really add your substrate solution and incubate it in the dark. This, because it's a colored reaction, this can be really sensitive to light. So some kits will request different timings. Some kits will want this at room temperature or at 37 degrees. So again, make sure that you are reading the product data sheet for this. But whatever you do, try and avoid light. So you can either put the plate in a drawer or you can have it out on the bench under some tin foil. Once your most diluted standards start to show some color, you want to stop the reaction with acidic stop solution. So in this step is also where you're going to see your blue product change to a yellow color. And importantly, once you stop the reaction, you really need to make sure that that whole reaction is stopped very rapidly. So once you've added in the stop solution, place that on a shaker for 30 seconds or for a minute and make sure the whole plate is nice and stopped. However, do be careful because this is an acidic stop solution. You really want to avoid getting it, obviously, on your skin or in your eyes. So for this step, definitely make sure that you've got all the correct PPE on. Probably wear a pair of lab glasses as well. And after you've stopped the solution, you really want to record your plate as quickly as possible. You don't want it to be sitting around for even really half an hour. You want to be getting that straight on the microplate reader. I always like to say to people that in our lab, we had a microplate reader that was definitely older than I was, possibly older than my supervisor. So it took a while to warm up. So we'd always be making sure that the microplate reader was up and running, at least before we'd put the substrate solution on, so it was ready to go at this time of the protocol. Make sure that you are using a suitable wavelength for your substrate product. So often, as I say, if you're using TMB, that the suitable wavelength will be 450 nanometers. But again, don't just assume this. Make sure you do know the exact reagents that you're using, because this could not pick up that color. And at this point is where you can think about your wavelength correction. So what we mean by that is when you come to reading your plate, if you also record the plate at a second wavelength that is outside of the limits of detection of that substrate, basically what you're getting there is a negative background. So for TMB, if you record it at a wavelength of somewhere between 570 or 650, that's just going to pick up any optical interferences that are caused by scratches or fingerprints or any other matter that isn't specific to the substrate solution. You can then basically use that as a background recording, and you can subtract that off of your other wavelength recording, and that's just a way of making sure, again, it's just another control you can use throughout. So throughout this protocol, there are many areas that you can introduce human error and affect the reproducibility of your assay. So if you haven't done an ELISA before and you've just seen this for the first time, or if you have done ELISAs, I mean, they're fairly straightforward experiments in terms of what steps to do. And as I say, they're quite nice in throughput. I really enjoyed doing ELISAs because the amount of data that you get out of them so quickly. But as I say, the one area that you really can't overestimate is just how easy it is to make this unreliable. And again, it all comes down to the fact that this is just a chemical reaction. So exactly what I just said, really. The speed of the reaction is going to be influenced by anything that affects the chemical reaction. So that includes temperature, binding kinetics, contamination, consistency, pipetting technique. If any of these things change, then you are going to get significant variation between each plate. And that's also why it's important to run your standards on every plate. And sometimes this is a bit of a silly suggestion, but one thing that you really should be aware of is like just the temperature that of the space that you're working in. So for example, I used to work in a lab that would have quite bad temperature fluctuations with the seasons. So if I was doing an ELISA in the middle of winter versus an ELISA in the middle of summer, the rate of those reactions would be completely different. And these are the kind of variations in lab environment that you just need to be aware of, and that you can jot down in your lab notebook, and you can just keep yourself nice and familiar with different things that could lead to possibly abnormal results. Also with the temperature, you want to make sure that you've warmed all reagents to room temperature before starting. So this is going to take a minimum of 30 minutes. So when you come in to your lab first thing in the morning, make sure you're getting those reagents out, make sure they're nice and warm. Because as I say, if you start using ice cold buffers, then you're really going to make that reaction happen a lot slower than you'd expect. And again, it's really going to affect your results. I'm sure a lot of you will be very, very experienced with pipetting, and I really don't mean to be patronizing with this slide, but it does have to be mentioned as poor pipetting technique causes huge variability in results. And there are actually some ELISA-specific pipetting techniques that we do recommend. So first, we recommend that you pipette at an angle and that you make sure you're not touching the bottom of the well. If you do, what you might be doing is accidentally scratching off of the ELISA plate coating. You might be ripping off your whole antibody-antigen complex, and then you're not going to get any signal. We also really recommend that you use multi-channel pipettes for this wherever possible. This is, again, just to help reduce user error, but also to help speed up how quick you can pipette across a whole plate. Obviously, if you're doing 96 wells, that can take a while. If it's going to take you 5 minutes to pipette a whole plate, then again, you're going to be seeing differences between the first well and the last well. Make sure that you are using new tips for different samples and standards to avoid contamination. And make sure that you avoid bubbles wherever possible, because that can really affect your plate readout. A useful trick for avoiding bubbles is to do something called reverse pipetting. Basically, that's where you eject the pipette all the way down to the bottom, and then you bring it up to the first notch rather than fully expelling it. If you're unfamiliar with this, or there's no one in the lab who can show you, you can find some videos of this on YouTube. It's a really commonly used lab technique, but again, it can just really help avoid you ending up with bubbles in your wells. And just like I mentioned before, make sure that you pipette in the same order of samples every time, because then if it does take you a bit of time to pipette the whole plate, you will have the same length of time between the start of the plate and the end of the plate. Washing as well is another really important step throughout your ELISA, and poor washing is a common cause of high background. If it's possible, we do suggest that you use an automated plate washer. They're incredibly accurate. They increase assay reliability. If you go through a lot of ELISAs, do see if your lab can invest in one, or just ask around. There might be another lab in your building that has one that you can use. However, I do know that using an automated plate washer is a luxury that not everyone has. So if you are manually washing, then there are some other things that we recommend. So we recommend that you do add in a 30-second rest period after adding the wash buffer. It's like a mini 30-second incubation with wash, just before you replace the buffer and perform another round of washing. Also, make sure that you're tapping your plates really firmly on fresh, dry tissue to remove as much of the buffer as possible and help get rid of any bubbles. If you are using wash bottles as well, you can try and just cut off the top of that bottle tip, which are quite small, and that will just help avoid you accidentally blowing out any bubbles with that as well. However, it's also important to make sure that you don't leave your plates to dry in between washing. So even though we really do want you to firmly tap that plate, get rid of all wash buffer, move on very quickly to the next step. So again, this comes on to make sure that the next reagent is ready to go. You don't want to leave that plate sat around for 5 minutes whilst you're making it up if it hasn't got any wash buffer in it. So for incubations, what we just say is to make sure that you follow suggested protocols. This is going to vary depending on kits. As I say, some incubations will recommend you perform them at room temperature. Some will be at 37 degrees. Some will be shaking. Some will be in the dark. Just make sure that you are fully reading your protocol. Make sure that if it's a new kit, that you've read it all the way through and don't just assume it's the same across kits. We also do say that even though we can all be guilty of it at times, try not to use buffers and diluents from different kits. So if you finished one kit, don't just, or if you finished one reagent from one kit, don't just grab it from the next kit if it's from a different lot, because all of those kits will be extensively quality controlled and validated using that specific batch number. So if you start messing around with these, then again, you're just going to really limit the validation, that you're going to question the validation of your kit. You're also going to lead to inconsistencies between plates. Also, if you're using multiple plates at once, something to consider is try not to stack your plates during incubation. And this is because you can end up with something called edge effects. So this is where there'll be changes in temperature distribution between the middle and the edges of the plate. And what you might see is that the readings around the edge of the plate are very different to those in the middle. So if you are doing multiple plates, just make sure that they're next to each other instead of on top of each other. And finally, for this section, I just want to briefly talk to you about antibody pairs. So I'm not going to talk too much today about homemade versus commercial ELISA kits, but if you are developing your own ELISA, then the most crucial part of ELISA development are the choice of pairs that you're using. Things to take into consideration are the fact that each antibody needs to bind a different epitope on non-overlapping regions of your antigen, exactly as I mentioned earlier. However, the use of these two antibodies is going to significantly increase sensitivity and selectivity of the assay. Make sure that the antibodies that you're using have been well validated specifically for a sandwich ELISA, not just a different binding array. Make sure that you can see and inspect the original validation data from the supplier. And ideally, you want these pairs to have been validated together as well. You also should always be thinking about the clonality of your antibody. So just a quick recap, and apologies to anyone who's been with us all week. Hopefully the antibody clonality lesson is well and truly stuck in your head now. But just for anyone who does need a recap, so your polyclonal antibodies are antibodies that are derived directly from the serum of immunized animals. So as you can imagine, when you immunize an animal, the animal is going to produce lots and lots of antibodies against that antigen. And they're all going to bind different epitopes, different binding areas on the protein surface. This is really good in a way because it means that that polyclonal mixture is going to have a really high overall binding capacity. It also means it's going to be really good for binding things like rare species or slightly mutated species. However, as you can imagine, different animals are going to produce slightly different immune responses, and even the same animal is going to produce a slightly different immune response over time. Therefore, you do end up seeing some lot-to-lot variability between your polyclonals. This can be mediated by using a monoclonal antibody. So a monoclonal antibody is derived from a hybridoma made from a specific B cell clone hybridized with an immortalized myeloma cell. That hybridoma is then cultured and will continuously produce the exact same clone of antibodies over time. However, again, over time, these hybridomas can experience something called genetic drift, and you are going to start to see some slight differences in your monoclonal antibodies over a long period of time. So this is where we then have our recombinant antibodies, which are kind of considered to be the future of monoclonal development. So to create a recombinant antibody, that involves in vitro cloning of the IgG domains, and you put that into a suitable expression system. And that's going to result in the exact same clone of antibody being produced over time. And because it's genetically engineered, it's going to have unrivaled reproducibility, and it's going to completely eliminate the risk of genetic drift. Monoclonals and recombinants, however, only binds and recognize one very specific epitope on the protein target. This is obviously has a lot of benefits. It means it's going to have much higher specificity, much lower chance of off-target binding. However, if you, as I say, if you are looking at a diseased or slightly mutated species, you might slightly avoid capture of all of them. So if you're doing something like protein pull down, that's where we lean towards a polyclonal. When you're using two antibodies as well in your antibody pairs, you need to make sure that you're titrating both of them to find the optimal concentration. And the best way to do that is something called a checkerboard titration, which you can see there in that little diagram. And that's basically just where you perform a titration of one of the antibodies along one of the axis, and then you perform the secondary antibody dilution on the other axis. And that's going to hopefully find you the optimum dilution pairing for both of them. So at Proteintech, we manufacture all of our antibodies in-house, and we actually have the largest in-house portfolio of pre-matched antibody pairs. So we now have antibody pairs targeting over 3,600 targets. And one of the reasons we can develop these so quickly and why our portfolio is so big is because of what Amy mentioned at the start, our epitope mapping. Because we know exactly where our recombinant antibodies bind on the epitope surface, we can very quickly predict which of our antibodies are going to work well together in a pair, and then we can test those experimentally. And because of this, as I say, our antibody pair range is the biggest on the market. So these are created using our proprietary rabbit recombinant and mouse monoclonal technology. And as I say, they're all validated using in-house multiplex bead array technology, but we always also validate with sandwich ELISAs. So if we're selling them for a sandwich ELISA, then we'll always validate them also in a sandwich ELISA. And exactly what I just mentioned, because we create these with our recombinant technology, we have unrivaled batch-to-batch consistency, we're very easy to scale up, and we have security of supply in all of these pairs. If you are looking for an antibody pair or even an ELISA kit and you don't know where to begin on our website, this is what we've got wonderful Able AI here for that Amy introduced at the start. So just earlier today, I typed into Able, I need an antibody pair for a sandwich ELISA, IL-6 human serum. As you can see, I didn't even be bothered to write the full sentence. I just said, this is what I need. And Able, as always, was really helpful and said, here are the recommended IL-6 matched antibody pairs that are validated in human serum for you. And it gave me 2 potential options. It told me the exact capture and detection antibodies that I used, so I could go check those out individually, shared the linear range and what their key features were. I then had a look at both of these and I said to Abel, okay, but which one should I use? And it recommended one product over the other, and it gave me a specific reason as to why. And in this case, it suggested using the pair that had both of the recombinants in, exactly because of what I just mentioned about the benefits of having recombinant technology in terms of batch-to-batch consistency and specificity. So if you are looking for any reagent or any experimental advice, do check out Able AI. As I say, it can really save you a lot of time when it comes to hunting through products. And then the other thing I want to mention about building your own ELISA kit is how to validate that ELISA kit for that exact sample type. So as I've mentioned, it's really important wherever possible that if you are buying antibodies or buying ELISA kits, that you buy ones that have been validated for your exact sample type. And this is because the sample type and the sample matrix can really affect antibody binding. If you are, as I say, making it from scratch, or if you're using an ELISA kit that hasn't been validated in your sample type, let's say you're using quite a unique sample type, or there's not many on the market available for you, these are some in-house validation techniques that you can do to make sure that your ELISA kit is going to work perfectly in the exact condition, your exact experimental condition. So the first thing you can do is something called spike recovery. This is where you spike a known amount of protein, so protein standard, for example, into both your sample matrix and your sample diluent, and then you calculate the percentage recovery for each spiked aliquot. Because your sample will also contain endogenous amounts of protein, you're going to have to measure that too and subtract it from the spiked sample. So you're going to add a little bit of your protein standard into a sample diluent. You're also going to run, you're also going to add it into your sample. You're also going to just run your normal sample as normal, and then you're going to perform the equation that's on the screen there, which is much more simple than it looks. And basically what you want is the percentage recovery for that to fall between 80 and 120%. If it's outside of that margin, then what it's telling you is that everything that's inside that sample matrix, so anything that's in your sample that's not your protein of interest, is impacting the way that that antibody is binding. And actually, perhaps that kit or that antibody isn't suitable for your exact sample type. So the other thing that you can perform is very straightforward. It's just called linearity of dilution. So this is where you serially dilute your sample and measure it against the standard curve. The reason we suggest doing this is because some matrix interferences will only occur if the factor is present at high enough concentrations. So this linearity of dilution allows you to pinpoint the exact concentration at which the sample matrix starts interfering with your antibody. But if it does work and if it shows a linear dilution of results, then what that indicates is that, yes, your assay is flexible across a range of concentrations and it's validated in this sample type. So now we're just going to spend a little bit of time on data analysis because it's perhaps a little bit more complicated for ELISA than for some other assays. So once you've read your plate and you've got your absorbance readings, in order to understand your pipetting precision and how reproducible your data is, you need to calculate the standard deviation and the coefficient of variance. So CV values greater than about 15% could be caused by pipetting error, could be caused by contaminated plates or reagents, uneven temperatures, or even evaporation during incubation steps. And this is where it's going to suggest that your data may not be accurate. This is also why it can be useful to run triplicates, because if one of them is way off, then you can rule that out as an error and you can use the other two. Otherwise, if you're seeing results of over about 15% in your duplicates, you are just going to have to run the experiment again. And as I mentioned earlier, make sure that you are running a standard curve for each plate. Because, as I've mentioned, the plate is so variable, the rate of reaction is so variable depending on experimental conditions. You need to make sure that for every single experiment you run, that standard curve is accurate. So we use the OD values from the standards of known concentrations to create that standard curve. We then interpolate unknown values from the standard curve to work out real concentrations in our samples. So the standard curve should range from almost undetectable to a maximum signal detectable by that pair of antibodies. As I've mentioned, don't expand that range. The curve is limited by binding characteristics of the antibodies, and expanding outside of the range could end up being unreliable. When you're selecting which curve of best fit to make from your standard results, the R² value can be used as a goodness of fit measure for linear regression models. There's lots of different linear regression models available. The most simple is just straightforward linear regression. However, in biology, most real biological data doesn't fit a linear curve, and data can often become compressed at the lower points. One way to overcome this is to try something like a semi-log or a log-log plot. However, as you can see there on the screen, the R² value is still only showing about 0.97. At Proteintech, we recommend trying the 4-parameter logistic curve fit, otherwise called the 4PL curve. We think this is best for fitting data from a biological system. So this is a model which estimates 4 parameters that help the data fit the curve. And as you can see, when you use this, the R² value is much closer to 1. There is also a 5PL curve fit that some people prefer, some other manufacturers might recommend in their kits. However, at Proteintech, we think the 4PL system works really well. And if you're not sure which one to use, there's a really easy tip you can do just to check, and that's to backfit your standard curves. So this is where you use your standards to fit a standard curve, and then treat those standard values as unknowns and just interpolate the OD values from those standards back to the curve, and they should read very, very close to the expected values. And whichever curve gives you the result that reads closest to those values is probably the one that you should go for. So scientists at Proteintech have come up with our own completely free curve fitting software. There are many curve fitting softwares available out there, but a lot of them require subscription or aren't fit for purpose. So if you go to our website, or if you just scan that QR code, then you can simply paste in your OD data. You can select your plate layout, so select how many standards you have, what your standard concentration dilution series looks like, how many sample replications you had. And hopefully this will save you a lot of time in your data analysis. So the default setting for this curve fitting system is 4PL, as I mentioned. However, all of this is completely flexible, so you can adjust it and change it to however you want. And as I mentioned, it will give you this standard curve from which you can extrapolate your sample concentrations. So it will do that for you. You can just click download, or if you want to hover your mouse over the curve, you can see the concentration that matches each of the OD values exactly. So I appreciate that's quite a lot of things to bear in mind. So here is just a nice simple overview of all of the different stages of data analysis that you need to do for your ELISA experiments. So as I mentioned, make sure you calculate your replicate averages, work out the standard deviation and the coefficient of variance, make sure that your pipetting precision is correct. Remember to subtract background absorbance from all data points, so this will come from your blank reading. Generate a standard curve with the best fit. Extrapolate your sample concentration from those OD values. And really importantly, last thing to do, don't forget to multiply your value by your dilution factors. So you need to make sure that your sample concentration falls within your standard curve. If you're looking for a really high abundant protein, this might involve you needing, as I said, to dilute down your samples at the start. It's absolutely fine to do that. It's also fine to dilute your samples by different amounts. So if you have a treated and an untreated sample, and let's say your protein of interest is significantly higher in your treated sample, you can dilute that one and not the other one, as long as you remember and multiply it by the dilution factor correctly at the end. That's all fine. And so very quickly at the end, then I'll come on to some troubleshooting. So a big problem for some ELISA kits is that you're seeing much too high a signal and your sample readout is higher than your highest standard, or they're maxing out on the reader. So exactly what I just said, you can try diluting your samples in sample buffer. If you think that all of your samples and signal is becoming much too high a signal too quickly, then make sure that your substrate incubation is being carried out in the dark. It could just be that the light is affecting the color change of the reaction. It could be that you're not washing sufficiently. Could be that you're introducing contamination, either through not changing pipette tips or from reusing plate sealers. Or also you could be using the incorrect concentration of salt in your wash buffer. So if you're making your buffers from scratch, or if you're using them from a kit, but you're diluting them, just double check that you've got your buffer quantities and your dilutions correct. On the other hand of the scale, then you might have too weak of a signal. So this is where your analyte is falling below the detectable levels. So for this, you consider using a more sensitive ELISA, or you could try using a more sensitive detection system. It could be that your reagents were not at room temperature. If your reagents are too slow, remember I said that that's going to cause your experiment to go too slowly, and you just might not have enough signal development. Again, make sure that all of your dilutions are performed correctly. If you're seeing no signal at all, make sure that you are reading at the correct wavelength and that you do know the exact enzymes and enzyme conjugates that you're using in your experiment. It also could be that you've accidentally scratched off, as I said, that antibody-antigen complex with your pipette tip. So make sure that you're not accidentally scratching the bottom of those wells. I would say the biggest issue with ELISAs that people face is high variation between plates. And this all comes down to everything that I've tried to hit home about today. Make sure that you're pipetting correctly, make sure that you're washing efficiently and uniformly. You're not accidentally introducing contamination or bubbles. You're not stacking your plates during incubation, as that's going to introduce edge effects. And the most important thing that's going to cause changes in results, as I said, is that temperature. So make sure all of your reagents are at room temperature before starting the assay, and make sure that, as I said, you just keep a record of if there are any huge temperature fluctuations in your lab that day. And if you are suffering with a high background, again, this is where you want to make sure that your kit is validated for your specific matrix. So this means the sample type that you're using. You know, if you're using a kit that has only been validated in cell culture supernatant, and all of a sudden you're using human serum, there might be a lot of other proteins in that serum that are just interfering with that antibody binding. So this is where you want to be performing your spike and recovery, your linearity of dilution, or just trying to find a kit that has been validated specifically for your sample type. Make sure that you're blocking correctly, you're washing correctly. Again, you're not introducing contamination. And as always, make sure that the time, temperature, and incubations are being performed at the correct level. So in summary then, ELISAs, I think, are very simple to perform, but they do need consistency. And this is where the difficulty with ELISAs comes in. There are lots of best practice tips that I've covered today for accurate and reliable data. Don't just forget about these. As I say, we do recommend using that 4PL curve for your line of best fit, but make sure that you are testing this and you're trying out different curves and back using the known concentrations as standards to test this. And finally, although commercial kits are expensive, can be expensive or more expensive than making your own, They are so extensively validated. You're going to have really, really high levels of inter and intra-assay reproducibility that ultimately could save you money and time in the long run. And just coming off the back of that then, I just wanted to talk a little bit about the different types of ELISA kits we produce, because we actually have 3 different types of kits available. So our traditional Proteintech ELISA kits, we have over 1,000 of them available. As I mentioned, they've been extensively quality controlled and we've had lots of time in development. We always validate our ELISA kits in-house with natural samples. So we don't validate just on the standard alone or on a recombinant protein. We always test on relevant physiological samples. Our kits are provided in a strip plate format. So this means if you only have a couple of samples you need to try, you don't have to waste a whole plate. You can save the rest of the strips, as I said, and run them again on another day. We have direct tech support directly with our R&D scientists who can help with any optimization or troubleshooting that you need. All of the sample dilutions are predetermined, and we are up to 50% cheaper than some of our leading competitors. We also do have our humankind authenticine ELISA kits. So these are ELISA kits that have been generated using our HEC293 human cell express proteins. This means that they are really, really sensitive, and they're really good if you're using particularly low volume or low abundant analytes, or if you've got particularly precious samples, let's say from a patient sample that you don't get much of. Because they have the native glycosylation and PTMs that you'd expect to see from human proteins, as I say, they can be up to 1,000 times more sensitive than leading competitors. So exactly what I said in that troubleshooting side, if you're seeing a low signal, then you might just need to be trying a more sensitive kit. And finally, I just wanted to highlight our brand new Speedy one-step ELISA kits. So our Speedy kits are our one-step solution for rapid analyte detection. So these are made with antibody pairs featuring our FC0 backbone. So this is a backbone that's had the FC receptor binding site mutated, so they can no longer bind FC receptors. Sorry about that. These kits can provide results in less than 90 minutes. So if you compare that to a whole day ELISA or even an overnight incubation as well, if you have to make that yourself, you can really cut down a 2-day protocol into less than 2 hours. They have much reduced complexity. They have a single wash step. So again, you're really going to be reducing all of those times that you can introduce human error or pipetting error. And because of that, as well as the fact that we're using our recombinantly manufactured backbones, they have unparalleled precision and sensitivity. So these really are quite a new, exciting kit we've got out on the market. You can scan the QR code there to learn more about them, or do get in touch. And we've just got an overview of the workflow there. So as you can see, all you have to do is incubate your standard samples and antibody cocktail for an hour, do 4 short washes, and then you can go straight on to the development stage. So if you are in a very high throughput lab, or you want to do some high throughput experimental development, then this could be a way to really, really shorten down that workflow, as I say, whilst also increasing the sensitivity of your experiment. And then very finally, just to highlight all of the products I've spoke about today, we have our traditional ELISA kits, we have our high sensitivity authenticine ELISA kits, we have our Speedy ELISA kits, and as well we have, as I say, our huge portfolio of our matched antibody pairs. And with that, I just want to say thank you everyone for listening, and I'll hand back to Amy.
Thanks very much, Poppy. Wonderful as ever. I'm just going to share my screen for some final slides. Let me know if you can see them.
Yeah, all good.
Oh, good. Fantastic. So yeah, thank you so much, Poppy, for that wonderful webinar. There's been a lot of people chatting in the chat box, and we'll have a look through some of the questions in just a second. But I would just like to remind everyone that if you have signed up for this webinar with an email that is, you check regularly, you will get a recording of this webinar. You will get a certificate of attendance. Make sure to wait at least 5 days. Well, it can be up to 5 working days for you to receive those. And because you have attended this webinar today, you are eligible to use the promo code antibody25 for 25% off a full-size antibody. In terms of checking out more things from Proteintech, as I said, this is the final installment of our Immunoassay Week webinars, but all of our recordings of any of our webinars that we've run will be on our video hub. I've put the link in the chat, but you'll also receive it from this webinar too. We also have our online learning portal, so Poppy is also very instrumental in building courses that are online and available to you. They are self-paced and certified as well. So there are things like how to do Western blotting, statistics for the life sciences, image analysis using ImageJ, and more to come. So please do check out our learn.ptglab.com portal. And also, like I said, this is the final installment of the Immunoassay Applications Week, but every month we do run webinars that are freely and globally accessible. Just again, I'd like to thank everybody for attending, and thanks Poppy for your talk. If anybody has any questions, please do reach out to events@ptglab.com, or if you want to personally reach out to Poppy for some ELISA specific questions, her email is poppy@ptglab.com. And if you like today's event and you want to keep up to date with our online and in-person events, please do follow us on social media. So I'm going to stop sharing my screen and we're going to go through some questions. So there's been a few questions in the chat that are quite project specific. So I'm going to go more on the general side of questions. And if anybody's got any project specific questions, please do feel free to reach out to Poppy after the session. But the first question is, could you go through what edge effects are and just again, kind of repeat what edge effects are?
Yeah, absolutely. So edge effects aren't unique to ELISAs. You can see this in any kind of experiment when you're using a plate. And basically, what we're saying is that the wells on the very edge of the plate are going to undergo very slightly different experimental conditions than the middle of the plate. So in the case of stacking your plates in an ELISA, what can happen is the areas in the middle of the plate and the plates that are in the middle of the stack are just going to be much warmer. So you're going to end up having different temperatures across the plate. And because, as I've mentioned, the temperature will greatly impact the rate of the reaction, you might see a much stronger development inside the middle of the plate. However, as I said, this isn't unique to ELISAs. So we used to also suffer with edge effects if we were doing 96 well, for example, cell culture. What can happen is that the edges of the plate can have higher evaporation of liquid than in the middle of the plate. And sometimes the cells on the edge of the plate would just be a bit more stressed or would need more feeding more regularly. So the best way, if you're worried about that, to check if it's happening is just when you're doing your repeats. I know how nice it is to stick with the same plate layout because obviously it's familiar in your head and maybe that's how you think of it, like, oh, A1 is the control and A2 is the positive. But actually what you really want to be doing is changing the location of your controls and your samples in the plate every time. And then if they are experiencing different levels of temperature or evaporation or anything like that, then you will be able to see that in your replicates.
Fantastic. Thanks, Poppy. Another question, obviously at Proteintech we have lots of commercial kits, but if somebody was to make their own ELISA, what kind of clonality should they be thinking about for their capture and detection antibodies?
Yeah, so another really good question. So as I say, all of our matched antibody pairs are made with our monoclonals or our recombinant antibodies. And the reason for this is because they only bind one very specific epitope so that you know that you'll be able to find a detection antibody that will also bind a specific epitope, and the protein basically will always be in the exact same conformation. If you have a polyclonal on the bottom, what can happen is the protein can bind to different areas, and then the binding region of the detection might not always be available. So you might just really limit that interaction. So again, we're not saying that monoclonals or recombinants are better than polyclonals, but specifically when you're developing your antibody pairs, having antibodies that only bind the single epitope is generally the better way forward.
Perfect. So just on the same vein as sort of sensitivity of an ELISA, if somebody's struggling to detect their protein, are there any sort of major troubleshooting things that they should be doing?
Yeah, absolutely. So I guess the first kind of question is, are you struggling to detect it because it's a really low abundant analyte, or because, maybe it's not present at without being stimulated? And in which case, then what you want to do is try using either a more sensitive detection method. So as I say, you could use our Authenticine kits. One thing you can also do is you can really try and basically just concentrate it a bit. So for example, if you're using cell culture media and you've discovered that it's just too dilute, you can maybe half the amount of cell culture media that you're leaving your cells in. Obviously, don't do this for a long period of time because you might be stressing them out. But let's say you're only doing a 24-hour stimulation. If you normally do that stimulation in 1 ml, reduce it to 500 microliters, and then you've doubled that concentration. The other thing you want to do is, if it's not to do with the amount of analyte present and it's potentially just a experimental condition error, then you can try heating things up to 37 degrees. So if you're currently doing your incubations at room temperature, or you're currently doing just try doing your incubations maybe at 37 degrees. Make sure that all of your reagents have warmed up enough before you start. You can definitely try that. As I say, the temperature even of just like the lab makes a huge difference. So record what your room temperature is, because if the kit suggests room temperature, but you're actually doing that at 18 degrees, that might be a few degrees lower than what room temperature is meant by on that kit.
Fantastic. And I think we've probably got time for one more question. There was quite a few people who talked about reverse pipetting. If you could just describe how to do reverse pipetting again.
Yeah, I realise it's quite difficult to not show with a pipette. Amy, I might ask for you to help with this. But you basically, when you're pipetting, normally you'll go down to sort of the first, what would you call it? The first stop.
Yeah.
Then you go down to the first stop, and then you pipette up, and then you expel, and then up, and then expel. And you only go all the way down to the bottom when you want to get rid of the very last bit of everything. But what we're saying to do is, if you go all the way down to the second stop and pull up to that stop in the middle, then you'll get the same amount of liquid out, but you won't expel it out and get the bubble at the end. Amy, feel free to, I feel like that maybe wasn't the clearest. If you've got a better...
No, that was probably as clear as I could do it as well, but there are some really good links online to actually show you how to do it. But essentially, yeah, so instead of going down to the first nodule to aspire up, go down to the second to avoid bubbles. So there are a few more questions, but they are quite project specific. So what I'm going to do is encourage the people who ask those project specific questions to email our tech-euro@ptglab.com inbox, or Poppy at ptglab.com, and she can direct you to the best person to look at your issues. Please feel free to send your actual data set as well, and we can give you some really specific troubleshooting advice. But with that, I'd like to thank everybody for your attention for the whole of the webinar, and also for people who have joined us for every day of Immunoassay Applications Week. Thank you for joining us, and join us next year when we do it again, and also each month when we do our other webinars. And with that, I'm going to end the webinar for everyone, and hope you have a nice weekend. Thanks, everybody.
Thanks, everyone. Bye.